Hello, and welcome to part two of our lecture on immunology. And we're looking at chapters 17, and I believe this is chapter 18. So, in this lecture, we're going to talk about some of the different applications of immunology, ways that we can use what we know about immunology to help us diagnose disease, treat patients, and come up with different types of therapeutics. So before we get too far into applications, we need to talk a little bit about the concept of a vaccine. Vaccine is an application of immunology, and the idea behind a vaccine is to expose someone to a disease, to a form of the disease, like a modified form of a pathogen, in order to initiate an immune response. We want to initiate just not the innate immune system, but also the adaptive immune system to create memory cells, hopefully memory B cells, memory T cells. This is basically kind of faking it for the primary exposure so that our adaptive immune system will be completely activated, and should we be exposed to the real thing, to the actual disease pathogen itself, then we would be considered immune to it. Our immune system would be able to kick in really quickly. We wouldn't have to wait for all of the different activation components, and we would be able to neutralize that possible pathogen before it could ever take hold and cause a disease. Now, the idea of vaccination came about in the 18th century, so in the late 1700s, early 1800s. About I want to say 1796, something like that. A gentleman by the name of Edward Jenner noticed that milkmaids who were milkmaids that had contracted a disease known as cowpox were immune to smallpox. The cowpox virus is almost identical. It's very similar to the smallpox virus that was causing large outbreaks and deaths in the United Kingdom at the time. Edward Jenner was in northern England. Now, milkmaids were young girls that were hired by local farmers to milk the cows and provide the milk for the farmers' wives and leave the milk with the families. And they would go from farm to farm and milk these cattle. Now, there was an outbreak of cowpox around the same time that there was an outbreak of smallpox. And these milkmaids would contract the cowpox disease. And it would cause a very mild form of pox disease. They would be sick for maybe a week or so, and then they would be fine. With having been exposed to the cowpox, they developed an immunity to the actual smallpox virus. So Jenner came up with the idea of, instead of using smallpox to inoculate people, which is known as variolation, he came up with the idea of vaccination. Vaccination was using a modified form of an organism to instill resistance to the complete form of that organism later on. Now, this idea, well, camera there, off of what was variolation. Been around for used by the Chinese 15th and 16th. The dried scab, dry them, grind them into a very fine powder, and then blow small amounts of this powder up the nose of young children. The children would develop an extremely mild form of smallpox. They would survive it, but they would then have immunity to smallpox for the rest of their lives. But variolation was a dangerous prospect, and it had a pretty high mortality rate compared to the idea of vaccination. So vaccination saved more lives by introducing a modified form of the disease, as opposed to accidentally over-inoculating someone during a variolation procedure. Now, the idea of vaccine, of course, has been introduced into society for a long time, and immunologists then came up with, or actually epidemiologists came up with a concept or a theory known as herd immunity. Now, herd immunity, I'm sure many of you have heard about in the news lately. It's talked about in terms of the COVID vaccine. We're trying to instill herd immunity, and until the world and our own population establishes a strong herd immunity, COVID-19 is going to be an issue for many people, and we are going to be in some kind of modified form of quarantine until this herd immunity takes hold. Now, herd immunity results when the majority of a population is immune to a disease, and herd immunity quick herd immunity, instilled herd immunity, is done through vaccination procedures. So, in the vaccine world, if the majority of a population is vaccinated against a disease, they are able to prevent or inhibit the transmission of that disease to those who cannot be vaccinated. People who have immunosuppressive or autoimmune disorders are incapable of receiving vaccines for fear of their immune system overreacting or not being able to react at all. So we have to protect others in our society who uh, for some reason or other, are unable to receive a vaccine. So the majority of us receive vaccines, become immune to diseases, and that helps protect the society as a whole. Now, the idea of herd immunity comes from the concept or the observation of behavior in herd animals. In herd animals, for example, a group of horses, a herd of horses, if there's a really bad storm coming on, the horses will form a large circle, and in the center of the circle will be the youngest, the oldest, and the sickest. And so those are the animals that are unable to take care of themselves, but the entire herd surrounds them in order to protect them from the worst elements of the storm. So the concept of herd immunity is that the healthy of us that are capable of receiving vaccines will get them so that we stop the chain of transmission and hopefully help to protect not only ourselves but others around us. Now, a vaccine itself is a suspension of organisms, and it's usually what we call an attenuated or a whole agent. We have a couple of different types of vaccines that are used. The idea is to initiate a primary response against a benign form of the pathogen in order to activate the immune system. So there are different types of immunity that are acquired. Now, in acquired immunity, right, we're talking about our acquired immune system, adaptive immunity. We have naturally acquired immunity and artificially acquired immunity. Naturally acquired immunity can be active or passive. If it is an naturally acquired active immunity is what you get after having a disease. Once you get the disease, you develop an immune response against it. This is naturally acquired. You actually got sick. You had the illness, and it's acquired by an active infection. Naturally acquired passive immunity is what a child, an infant, has at birth. This is where the infant IgG antibodies will cross the placental barrier into the fetus, and the fetus, for a short period of time, for a few months, will actually have the same antibody panel as the mother. Now, the mother naturally acquired that immunity and then is passing that on to the child. Now, the child is not immune for the rest of their lives because of those antibodies, but they are temporarily protected while their own immune system is maturing and finishing development after birth. In artificially acquired immunity, we also have active and passive. So artificially acquired active immunity is acquired through vaccination. We are literally activating the immune system. We're just doing it with a vaccine instead of the actual illness itself. Artificially acquired passive immunity is when the antibodies from one source is introduced into another body in order to fight infections. A few years ago, when the United States had a few doctors and nurses that came back from Sierra Leone, and they had Ebola. They had contracted Ebola. There was two nurses that contracted Ebola, and they gave blood transfusions. The serum from their blood, what they did was they extracted antibodies from one patient and provided those antibodies to other patients that were not doing as well. So somebody had contracted the disease, and now they are giving those antibodies to someone else to help their immune to kind of fight the virus while their immune system is catching up. A recently used artificially acquired passive immunity is one of the therapeutics in COVID that has recently been granted FDA emergency approval, and that is an artificially acquired passive immunity. It's an antibody treatment that is being used. That is also the same treatment that the President of the United States received when he was suffering from COVID. Now, as far as vaccines go, we have different types of vaccines. We have what are called attenuated whole agent vaccines. These are modified forms of the pathogen, but the pathogen itself is intact. So the living organism or virus, active form of the virus is used. It is just altered so that it can't cause infection. So they call these weakened or attenuated vaccines. They are the one type of vaccine that will most closely resemble a true infection. So the closer we can get to an actual true infection, the greater the immune response, or what we refer to as efficacy, of the vaccine will be. Most vaccines that are of attenuated whole agent type will provide some kind of lifelong immunity. If not lifelong, it will be for tens of years. It'd be for an extremely long period of time, but it can be lifelong immunity if the patient is given boosters in wide increments throughout their life. A few examples of attenuated vaccines, so these are weakened forms of the pathogens, would be the Sabin polio, the MMR, typhoid, and TB. There are others out there, but those are some of the common uh, more common types of vaccines. Then we have inactivated whole agent vaccine. Inactivated whole agents are where we take the entire pathogen, but let's say it's a virus. The entire virus capsid and spikes would be present, but the genetic material inside of the virus would either be completely destroyed or removed. So these are killed microorganisms and inactivated viruses. Some examples of these include the Salk polio vaccine, which was the first vaccine to be introduced against polio, and the rabies vaccine for humans. Animals receive an actual attenuated whole agent rabies vaccine, whereas humans do not. Humans receive a inactivated whole agent vaccine, and humans only receive the rabies vaccine if they are suspected of being bit by a rabid animal. So we do not get a rabies vaccine. It's simply used as a therapeutic after the fact. Then we have toxoids. Toxoid vaccines are vaccines where it's not the pathogen that is actually causing the problem or that is the antigen, it's a toxin produced by it. The Tdap vaccine or DTaP is an example of this. Tetanus and diphtheria are both toxoid vaccines. So we initiate the immune response to create IgA and IgG antibodies against the actual toxins. So that if those bacteria, because these bacteria are incredibly common, and if these bacteria get into the system, we will have vaccines that will neutralize any toxins they might possibly produce. Toxoid vaccines don't have a real great efficacy rate, so they do require multiple doses, and that's why they're usually given in a series or with boosters. Then we have subunit vaccines. These subunit vaccines are antigenic fragments. This is where you would receive a piece of it, such as a viral spike or an antigenic determinant. These can be produced through genetic modification or through recombinant DNA technology. They're sometimes referred to as a recombinance vaccine. The other two types of vaccines are the conjugated and nucleic acids. If you remember back in adaptive immunity, B cells are where we get our memory cells for antibody production, and these are really, that humoral immunity is a key component of vaccinations. But if you remember, activation of B cells in this process can be dependent or independent. And in independent B cell activation, what we have are usually polysaccharide antigenic determinants. Our immune system does not recognize carbohydrates very well, and because it's a large polysaccharide antigen, we don't get the stimulation from T cells to create memory cells. So this would kind of defeat the whole purpose of the vaccine to begin with. So with some organisms, their antigens are of a polysaccharide formation, so we conjugate them, we attach them to a protein. There's only one or two different proteins that are approved for this, but they're conjugated to a protein, so the protein will initiate and take it from being a T-independent antigen to being a T-dependent antigen. These types of vaccines are oftentimes used for small children, newborns, infants, who their immune system is not fully developed yet. They can sometimes be used for people coming out of immunosuppressive disorders. The Haemophilus influenza vaccine is an example of a conjugated vaccine. The pneumococcal, streptococcal pneumonia vaccine is also conjugated. The last are the DNA vaccines or nucleic acid vaccines. This is where small plasmids containing the gene for the antigenic determinant are injected into muscle tissue. The muscle tissue will uptake this piece of plasmid and incorporate it into the DNA so that the host will have a small number of cells that are going to constantly produce the antigenic determinant. This will in turn be a self-boosting vaccine. The person would be boosted throughout their entire lives. This is not in use in humans yet. This is only in use that I am aware of at this point in time in horses for for an encephalitic disease here in the United States. It's the only DNA vaccine in the US. There's another DNA vaccine. It's used in Canada. It is not used here in the United States, and it's used in salmon farms for salmon, but it's not used in humans. Just a real quick look at some of the principal vaccines used here in the United States. We use these to prevent bacterial diseases. This includes the DTaP vaccine, which is diphtheria, pertussis, and tetanus. Notice diphtheria and tetanus are both toxoid vaccines, whereas pertussis is an acellular fragment. The DTaP vaccine is a multivalent vaccine. Multivalent simply means that we are giving more than one antigen. And in this case, in DTaP, it's trivalent. We're vaccinating against 3 different pathogens. Then we have meningococcal meningitis. This is a polysaccharide, so this is going to be a conjugated vaccine. Haemophilus influenza, another conjugated, as well as the strep pneumonia. Here we have some common virus vaccines: smallpox, polio, rabies, hepatitis, flu, measles, mumps, rubella. This is your trivalent MMR vaccine. The chickenpox vaccine, and you can see, and of course, hepatitis B. Now you can see for most of these are going inactivated would be an attenuated agent. Here's more inactivated would be a destroyed agent. Attenuated would be modified. The polio vaccine, by the way, poliomyelitis. Smallpox has been eradicated. There's only copies of smallpox in Russia, the CDC in Atlanta, and I think one other disease research center in Europe. Polio was the next virus that was supposed to be eradicated from the planet, like smallpox was. But one of the problems with polio is there are 2 vaccines, right? There's the Sabin and the Salk. Now, the Salk vaccine was what Jonas Salk introduced in the 40s. It was the first vaccine against polio. It was donated. He made no money on it. He gave it away for free, and it was funded by the March of Dimes. It was a very effective vaccine, but it was had a little bit of trouble in distribution. Now, a little bit later on, they wanted to instill greater immunity, so the Salk vaccine is inactivated. The Sabin polio vaccine is attenuated. Okay? So what they did was they modified the DNA inside the virus in the Sabin vaccine so that the virus was able to gain entry into cells and start the replication process, and this introduced more antigens and had greater efficacy later on. But the problem is sometimes the Sabin polio vaccine, because it was attenuated and not inactivated, would revert back to a wild type polio. And this started the spread of a disease known as VAPM. VAPM stands for vaccine acquired poliomyelitis. And so this was where people developed polio because they were given just the Sabin vaccine. Now, to prevent the induction of the VAPM, because once somebody got this, they became a spreader. They could then transmit wild-type polio to people around them. So in this case, now the Sabin and Salk vaccine are given together, so that should the Sabin, and this happens extremely rarely, but if the Sabin vaccine does revert back to wild type, the Salk vaccine will have initiated enough of an immune response to prevent that person from actually contracting polio. In diagnostic immunology, here, this type of application is where we really just use antibodies. And remember, antibodies are highly specific about what they attach to, so antibodies are going to be used in diagnostic testing. In diagnostic testing, we have two terms we want to pay attention to: sensitivity and specificity. In sensitivity, this is the probability a test will be reactive if the specimen is truly positive. And specificity is that it will not react if the specimen is a true negative. Now, an example of a test to explain, and kind of this is a hair splitting difference, you might think, but it does make a difference, is the Koch TB experiment and TB test. So this is a TB test here on the skin. They prick the skin with the antigenic determinants for tuberculosis. If you have been exposed to tuberculosis or had a vaccine or have survived tuberculosis, you're going to pop up positive on a Koch TB test. Because you have antibodies against it, and the scratching with the TB antigen inflicts an inflammatory response because your immune system is responding. And your test has to be read within 24 hours. Now, this works because this test has really high specificity. Okay, it's only going to react against the TB antigen, and only antibodies against that TB antigen will cause the inflammatory response or feed into that inflammation. So this test has really high specificity. We're not going to have a positive reaction if somebody has another disease that might even be closely related to TB. It's only going to react if it's TB, so it's very specific. The problem with the TB test, however, with the skin test is sensitivity. Yes, it's a true positive. Yes, antibodies are present, but what we don't know is if those antibodies are due to acute infection, previous infection, vaccination, or simple exposure. And so it is sensitive in that it will react, but the problem is we don't know what that true positive is. Is it vaccine? Is it exposure? Is it a previous infection? This is why no matter what, even if you have paperwork showing that you had a vaccine a month ago, if you pop up positive on a TB test, they're going to send you to the hospital or to a clinic to get x-rayed. They're going to do chest x-rays. But antibodies have really great specificity. They're super specific, and they're pretty darn sensitive, really. So antibody specificity is a really great base for developing immunological testing. Now, where do we get all of these antibodies from? That's the one big question. I know when I was learning about some of these applications, I was like, are they extracting them from other people? Where are they all coming from? And what is used now are called monoclonal antibodies, or MABs. Now, MABs are the result of hybridization between immature B cells found in the spleen of mice and cancerous B cells from cancer cell lines. These are used together for long-term antibody production. Cancer cells don't really die very easily, so we can keep them around for a long time. We need immature B cells because the immature B cells have not been initiated yet, so we can fuse the two together and create what's called a hybridoma. Hybridomas will be activated against our antigen of choice, and they are going to be highly specific. They will produce antibodies for an extremely long period of time, and all of the antibodies will have uniform conformation, so they'll be all producing the same antibody. Over here on the left is just kind of a little diagram walking you through it. So we take spleen from mice, we put it in a suspension that separates the cells from each other. We add to it some of those cancerous B cells, myeloma cells, and these two types of cells are brought together in a test tube. They're mixed, certain enzymes are added to kind of separate everything and allow the cells to fuse together. They will fuse together, be exposed to an antigen, and then placed into small wells. It's called seeding. They'll be seeded. The wells will be seeded, and if we have a hybridoma cell, because these are cancer cells, the cells will grow in these large clumps. If I see these large clumps here, then I know that I have hybridized hybridoma cells. If I have cells that are simply just growing in a single layer on the bottom of my wells, then I know that those cells have not been fused. They are not the cancerous, they are unfused cells. I then expose these to my antigen, and now my hybridoma B cells, immature B cells, these hybridomas will now produce antibody. They'll be activated against whatever antigen it is that I introduce. So I can create antibodies this way against any possible antigen that I want to. So what do we do with all of these antibodies that we're making? Well, one thing that's done with them commercially is the pregnancy test, the home pregnancy test. The home pregnancy test is a glorified form of a test we call an ELISA. And we're going to talk about ELISAs in a minute. So monoclonal antibodies have been created in the laboratory against HCG, the human chorionic gonadotropin hormone. This is a hormone that women only produce when pregnant, and it is excreted in urine. So this is why you pee on a stick if you are testing if you are pregnant or not. One of the windows, the test window here, has an antibody in it. When the HCG protein binds to that antibody, a secondary antibody is also present that has an enzyme on it that will change color in the presence of the urine. So if this antibody antigen complex is made, we get this sandwich formation. We talked about precipitation reactions in laboratory. This is similar to the titer test that is done in lab. Here we are going to be looking at the soluble antigen. Either patient antibodies, patient serum is oftentimes placed on top of a thin or modified agar solution containing antibodies against a specific antigen. If your patient serum contains that antigen, a zone of equivalence will form, and we will see this precipitate right here in the middle. This is sometimes referred to as a ring test. Or you can also call it immunodiffusion, just like we did the titer. But it's called a ring test because it forms this ring in the center. Now, you could have known antibodies in the bottom and place patient serum, or you could have patient antibodies in the bottom and add serum that contains different antigens to see if that's what the patient's antibodies are against. Agglutination reactions, this is very similar to the CRP test that we discussed in lab. Here, instead of soluble antigens, we're looking at particulate antigens. This will include circulatory IgM, which are really big, giant antigens, and we are looking for agglutination reactions here, because remember, these are particulates, not soluble antigens. Now, we have two different types of agglutination reactions. We have direct agglutination, where we are looking, this here is direct agglutination, where the antibodies are acting directly with the bacteria or pathogen that they are made against, and that those antibodies are in the actual body that is being tested. So we're looking for patient antibodies. Indirect agglutination is when we take antibodies from another source to test for, say, a protein of our own. So we're looking for a host antigen or autoantigen. The CRP test that we did in lab, that was an indirect form of agglutination testing. Then we have a typical antibody titer, a hemagglutination test. And in the hemagglutinin test, this is testing different concentrations of antibodies against a specific antigen. Now, antibodies from the patient, their serum is going to be put aside in different dilutions, just like we did in the other lab. So here's a 1 to 20, 40, 80, 60, 320, 640. Antibody reactions can be from a current infection, or is it from existing immunity? If I have somebody who has a really super high titer, like a 640, then they probably are in a current acute phase of infection. But if their titer is really much lower, then that's telling me it's either memory immunity from previous infections or they've gotten over it. If someone's titer rises significantly over a short period of time, that's indicative of early stage diseases. IgM titers are used to check for acute infection. This here is an IgM titer that we're looking at here, and it's done with blood cells. So what happens is they look for specific antibodies, and they add blood to these antibodies. Now, if agglutination occurs, these blood cells are going to be spread out, and we get this wide red color right here. So this is positive for agglutination. The way that these little wells are formed, there's like a little conical tube at the bottom, like a little divot. And if there's no agglutination, the red cells will sink to the bottom and form this circle like this, and they all just kind of settle on top of each other. They're not spread out because there's no IgM, circulatory IgM antibodies spreading them out, and so this is negative for agglutination. And that brings us to our final test here in applied immunology, and that is enzyme-linked immunosorbent assays, or ELISAs. There are two types of ELISAs, a direct ELISA and an indirect ELISA. The direct ELISA is used to look for the presence of antigen in a patient serum or patient sample. Now, before we get into the close-up of this test, I just kind of want to show you down here. This is what a ELISA test looks like, what the plate looks like. And so we're going to take one of these wells here and we're going to blow it up and take a look at it. That's what we're looking at here, is one of these tiny little wells. Now, most ELISA tests are commercial kits. So in the laboratory, when they create an ELISA, when you order it, you get the plate. The plate will already have the antibody in it. So in those wells, the bottom and sides of the wells are actually coated with antibodies, and they're attached to the well by their FC constant region. This is so the antigen binding sites are sticking up, waiting for antigen to be exposed. We take patient serum. The red blood cells, RBCs, are spun out of the patient serum. If the patient has the antigen, when the serum is added, the patient sample is added. Now, this could be serum, this could be urine, it could be saliva, it could be, wherever we think this pathogen may be present. The patient sample is added, and if the bacteria or if the pathogen is present, the antigen is present, then it's going to attach to those antibodies. Now, we're only showing one antibody in this illustration here, but remember the bottom of this well is coated with antibodies. So multiple copies of that antigen will be attached to multiple antibodies. We then wash out this well to wash away anything that might be left behind that's not attached to an antibody, and we add a second solution. The second solution contains a second antibody, and this antibody will be effective against the same antigenic determinants that the primary antibody is. This is called the primary, and this is the secondary. The secondary antibody, in addition to attaching to the same determinant, will also contain, on its constant region, it will contain an enzyme. That enzyme, how they attach them to the FC region, I'm not really sure, but that enzyme is attached to the constant region of that secondary enzyme, and it's sticking up. Finally, that will get washed out to remove any unbound antibody, secondary antibody, and a third solution containing substrate for the enzyme is then added to the well. The substrate reacts with the enzyme, and the reaction results in a color change to the fluid inside of the well. So if we have a visible color change, this is going to tell us that the serum from the patient contains the antigen that this ELISA test is specific for. ELISAs are great. They're really rapid. They take anywhere from an hour to 2 hours to run, so they don't take a long time. They're commercially prepared. They're very easy to do, easy to interpret. They're used a lot in drug testing. So this would be done with urine samples to test for metabolites of drugs such as cocaine, heroin, opiates, painkillers, those sorts of things. Here's another picture of a direct ELISA, and of course it's called direct because we are looking to see if the patient contains the actual antigen. Now here is an indirect ELISA, and an indirect ELISA, instead of detecting antigens in our patient serum, we're just reversing it, and we're going to detect the presence of antibodies. So in this case, instead of antibodies being adhered to the bottom of the well when you get it, it's going to be the actual antigen itself. So the whole bottom of the well is covered in antigen. We add the patient serum. If the patient serum contains antibodies against that specific antigen, they're going to stick to the antigen. We wash those out, and then we add a monoclonal antibody called an anti-HISG. This is the human immunoglobulin that's going to it will bind to the constant region of IgG antibodies. So it'll bind to it'll bind to the constant region of any antibody. So it binds to the antibody. These are monoclonal antibodies. They're man-made. They will bind to the constant region of that antibody. So now I have an antibody bound to an antibody. This is my primary. This is my secondary. Now this secondary antibody will have enzyme attached to it as well. So what do? We wash out any antibody not bound, and then we add our final solution with our substrate. The substrate will react with the enzyme, causing a color change within the well. An example of the most commonly used indirect ELISA test is to test for HIV. In order for someone to test positive for HIV, they have to have 3 out of 5 antibodies. I think it's 3 out of, well, this is 3 out of 4. I believe it's 3 out of 5 now. So again, here is our indirect. We're using antigen to detect antibodies, and this is an HIV test. This is our positive control. This is our negative control, and these are the 4 different antigens being tested for it. This patient tested positive for 3 out of 4, which is considered a positive HIV test. If you notice, the color change here gets slightly lighter. This is because the test is run in a serial dilution. This gives a general idea of which antibody is the most prevalent in this particular infection. All right, we already covered disorders associated with the immune system. I did this in another lecture. That recording is already up for you. So I just wanted to kind of finish up 18 with some of the applications of immunology that you're going to be tested on in the exam. So if you have any questions, feel free to let me know, and I will see you in class.